My name is Mike Ritter. I'm one of the founders and the CEO of Slant Range. We are a San Diego-based remote sensing and data sciences company. Our solution basically quantifies the performance of crops. And our client base are primarily input suppliers, crop science companies, and they use our solution within their field trials. So today I wanted to basically run through two things with you. One is to kind of introduce you to some of the methods that we're currently using, and then give you a little bit of an introduction to where this type of technology is going longer term. So a quick overview of what we actually do from a solution point of view. We're really involved at all stages from the planning to the data delivery. So we sit down with our clients and try to understand what their needs are in terms of how can we quantify a crop's performance to help them solve whatever questions they're trying to answer. That starts from the planning stages when we map out what are the data types, what are the traits that we want to measure, what stages of crop development do those measurements need to be done, what data types do we need to source. During the data collection stages, we oversee data from whether we're pulling in third-party data from satellites, for example, to overseeing third parties who might be using our own remote sensing technologies to pull in data from low-altitude robotic aircraft, to pulling in our clients' own data from some of their instrumented implements. We have an edge analytics app, which is basically a tool for use at the field edge that's used for scouting purposes, but also it's an important data QA tool to make sure that the data that we needed to get on a particular day was gotten. And then really the central point of it all is a cloud analytics package where we do the aggregation of data from hundreds or thousands of trials, wherever they might be distributed, aggregate all that data, extract the traits, develop models and forecasting tools. And then we also have some sharing capabilities built in, so APIs to enable push-pull with other data platforms. And then we do a lot of customizations depending on what our client needs are. The enthusiasm for this has just been huge over the last couple of years. We introduced the capability initially in 2017, and since then our clients have analyzed over 8 million plots in over 1,000 trial locations with this system. We operate primarily in 9 major crops. There's a handful of smaller ones that we've worked in, and deliver regularly 17 different types of traits or data products. And this solution is backed by a pretty robust portfolio of patents. So key question is, why is anybody interested in this particular solution? First and foremost, it's about automating and standardizing practices that have traditionally been done manually, that are subject to error and subjectivity in measurement. Second, the data that we supply provides our clients a real evidence of efficacy of their inputs, which enables ultimately faster, lower risk, lower cost time to market for new inputs. And then the data that we're collecting is ideally suited to generate new models to predict how inputs will perform in different growing environments. So the first question we always get is, well, can we automate, can we standardize some of the practices that we've been doing forever in a manual sense? And machine-based measurement offers a lot of advantages. It's faster, it can be more accurate, very much more sensitive than human observations. We can do measurements that humans might not be able to do, and it's more repeatable. It's software doing these things. It's not a human whose opinions might change day to day. So those all add up to a more automated and standardized solution that reduces error, and it removes subjectivity in the measurements, which ultimately improves the statistical confidence suppliers need in order to advance different products in their pipelines, which ultimately reduces the time and cost to do so. And it's important to note also that all this data that's being collected provides a permanent record of how a trial performed throughout the course of a season that can be returned to later in time if needed. We also shouldn't overlook that these tools provide some management capabilities. So data wrangling, data management is a nightmare for anybody who's really gotten involved in this type of thing. So with a platform like this, all of your trial data is basically organized into a single place where there's no more manual image processing by any of your own people. There's no more aligning images to plot patterns. That's all automated in the platform. And essentially no more data management headaches. So the key point here is that for every plot and every trial, it's archived, it's cataloged, and it's immediately accessible by whoever on your team might be interested in using that data however they will. So I want to run through a couple of examples here briefly before I close up. This is kind of a screenshot of our cloud-based platform. But before I jump into that, I'll say that the real enabling capability here is high-resolution spectral imaging. What I mean by that, when I say high resolution, I say high spatial resolution, meaning we are able to resolve the size and the shape of individual leaves. High spectral resolution, meaning we can change or we can detect subtle changes in pigment concentration in the leaf canopy that give evidence of changing health that a human might not be able to pick up at all. And then a high temporal resolution, meaning there's a revisit rate where we can actually map out the response function of a given trait and to different changing environment conditions. So this particular example, quick orientation on what you see on the screen here. On the left is it basically just an aerial view of, this happens to be a canola trial. It's an aerial view of the canola trial on this one particular date. At the bottom right is a high resolution or kind of a zoomed in window of that picture on the right, on the left. And on the upper right are all the dates that this particular trial was measured. Now, on the image on the left, there's actually a heat map overlay on that. And what the colors are indicating are the fraction of the plot that's in flower on that particular date. So at the bottom left, there's a histogram, and that basically summarizes for all of the plots on that particular date, what was the percent of that plot that was in flower. And then there's a yellow line chart at the middle at the bottom, and that is for a single plot that's been selected, and that's indicated by the white box up there. How did the flowering develop for that particular plot over time? So for canola seed suppliers, for canola producers, the ability to predict flowering time is important for application of inputs. It's related to yield, but anyway, this is a quick example of how canola breeders are using this application. A second application to show here is, this happens to be a soybean trial. It's laid out just like the last one was. Here, what we're interested in is measuring maturity. So what we're seeing in the heat map overlay on the left side of the image is actually days to maturity for all of the plots. So the histogram at bottom left is actually the days to maturity for every plot that in that particular trial. And it's hard to see the numbers there, but they kind of range between 110 and 140 days, I think. The yellow line is how we're mapping out the development and senescence of a single plot over time here. So the products that we're delivering ultimately, days to maturity, the date of maturity for a plot, and maturity ratings more specifically. And then next season we're excited to be taking the similar capability early season and looking at what we call vigor velocity, which is a way of saying how aggressively did those plants leap up out of the soil after planting. The last example I want to show here is around demonstrating input efficacy, and this happens to be a corn trial. And one of the difficulties with proving efficacy is that given the myriad variables at work over the season, how do you attribute performance to any particular management action or input or protocol? And so if we're able to zoom in, so to speak, on a specific trait over a specific window of time, and in this particular case, looking at chlorophyll concentration or canopy structure, and see how those traits evolve in response to a managed stress introduction or in response to a particular protocol of treatment, we can actually get a much closer picture of how that particular input or translated into plant response function. And so this is much more direct evidence of efficacy for inputs. So just my last slide then in closing up here, which is to look a little bit more to the future. Historically, our ability to predict how a crop will perform has been limited by our ability to measure how it is performing today. We've been limited in the types of traits we can measure. We've been limited in accuracy and the comprehensiveness of what we're able to measure. We've been limited in the standardization aspect in comparing measurements over time and geography. So if we are able to measure key performance indicators that have a greater correlation to yield or whatever variable you might be interested in, if we can do that with greater accuracy and sensitivity, and if we can do that in a standardized method so that we can compare results over time and geography, we have much better tools to actually predict how an input or a protocol is going to perform in any particular given environment. And once you've got that, now you've got tools to help prescribe how a grower should use those inputs in the future. So that's a quick summary. And if you're interested in learning a little bit more, we've got a booth right over here. Thank you.